Hello and welcome. In past couple of videos, I have been talking about parasexual behavior in prokaryotes. This is one ways by which prokaryotes show variability among themselves along with mutations. I talked about uh, two processes, that is transformation and conjugation, where foreign DNA is transferred from, say, outside to inside the cell in transformation, and by cell-to-cell contact between donor to recipient cell in process of conjugation. There is another third process which is involved in parasexual behavior, that is transduction. Today, I am going to talk about this process of transduction. We will learn about what this process is and how it takes place, and what are its application to molecular genetics in terms of gene mapping. Because like other two, transformation and conjugation, this process has also been exploited for mapping of genes on the bacterial chromosome. So before I proceed, a request: if you have not subscribed my channel, please do so, and also hit the bell icon so that you get the announcement for my future videos. Okay, so let us begin by defining what is transduction. As I mentioned earlier, here the DNA transfer from donor to recipient takes place by way of or through bacteriophage. Now, what is this bacteriophage which transfers DNA from donor to recipient cell? Bacteriophage is a virus which survives on bacteria, or in other words, which completes its life cycle inside bacteria, and while doing so, it kills the bacteria and the progeny viruses are released, and they infect other cells after that. So basically, bacteriophage, we are talking about viruses which survive on bacteria. So here, the transfer of DNA from donor to recipient takes place through these viruses, which we call bacteriophages. So before I proceed, I will very briefly explain what these bacteriophages are and how they complete their life cycle. So basically, host cell gets infected by these viruses. For example, T4 bacteriophages. We have lambdaphages. We have examples I'm giving. So this whole thing starts with the attachment of this virus, which looks like this under electron microscope. And the first try to attach on the host cell with the help of these structures. And this attachment is irreversible. Once the attachment is made, then the virus cannot be, or bacteriophage cannot be removed from the host cell. Okay, and then there is contraction between this sheath, and the nucleic acid is injected into the host cell, and that results into DNA uptake. That is, DNA of the virus enters into the recipient cell. Okay, that's how this infection of virus takes place into bacterial cell. first by it attaches itself, and then there is a contraction at this point, and then the nucleic acid is injected into the cell, and the DNA is released into the recipient cell. Now, there are two types of bacteriophages based on how they complete their life cycle inside the bacteria. One is lytic or virulent type of phages. Here, what happens once the DNA is injected into the cell, host cell, it results into the lysis of the cell, and it releases the progeniphages. So so means once the DNA enters, it multiplies, it makes copies of its own genome, and then it is packaged, and the host cell lysed, and the progenivirus is released, like T4 bacteriophage, for example. And another class of bacteriophage or another type of bacteriophage is the lysogenic, okay, or temperate phages. Now, here what happens, once the DNA, the phage DNA is injected into the cell, instead of multiplying, it enters into the host chromosome. It gets integrated into the host chromosome, and then it lies there in the host chromosome in a quiescent state. In the sense, it won't do anything until unless it is induced to come out of the host chromosome and undergoes multiplication, and ultimately cell lysis takes place, host cell lysis takes place, and the progeniviruses are released. So basically, bacteriophages are of two types. One is lytic or virulent, and another is lysogenic. And the difference in the two is, in the virulent type, the bacteriophage immediately multiplies and kills the cell, and progeniviruses are released. Whereas in lysogenic bacteriophages, it gets integrated into the host chromosome and lies there. And this, he can lie, the bacteriophage can lie in this cell for generations. Nothing will happen. It will multiply along with host chromosomal DNA, and it will go to the daughter cells, until and unless a situation comes whereby it comes out of the host chromosome and multiply, and it completes its life cycle by multiplication and then packaging, and the progeniviruses or phages are released. Okay, so these are two types of phages. Okay. Now, what happens? The question is how it remains quiescent in the host chromosome. So what happens? There is the expression of the viral genes remains repressed. None of the viral gene gets expressed so long it remains in the host chromosome. But then what happens, which induces the lytic cycle in these lysogenic cycle in these lytic bacteriophages? So what happens when the host cell is going to die, or its DNA gets damaged beyond repair, and the phage realizes that the cell is going to die, the host cell is going to die, then it comes out of the host cell and then multiply itself, and the progeniviruses are released. So phage is very intelligent in this case, that so long host cell is healthy and it is multiplying, it is lying silently there in the host cell. But once the host cell gets damaged, its DNA gets damaged, and the phage realizes that it is going to die, then it comes out of the host cell and completes the. So this is what happens. So let me explain to you with the help of cartoon what I'm talking about. So for example, this is the host cell, and the virus is injected into it, into this. So how the lytic cycle takes place? The DNA enters here, it multiplies. It multiplies like the example is from lambda. lambda phage, it shows both kinds of life cycles, lytic as well as lysogenic life cycle. And see, the host chromosomal DNA is breakdown, and then, this is packaged, and the progeny, and I mean, it gets, this DNA gets assembled into lambda phage, and the progeny phage is released, and this host cell gets lysed. But in the lysogenic life cycle, what happens when this DNA is injected into it? It gets integrated into the host chromosome, and it remains there, as I mentioned, and it multiplies along with the host cell. Generation after generation, nothing will happen. But then the when the host cell gets damaged, as I was mentioning, the induction of lytic cycle takes place, and as a consequence, this comes out, the phage DNA comes out of the host chromosome, and then it enters into the lytic cycle and follows the same. It multiplies, the phage DNA multiplies like this and gets packaged, and the progeny viruses are released. So that's how these phages live or complete their life cycle in bacteria. Now, we started with talking about transduction, wherein the donor DNA is transferred from donor cell to recipient cell by way of phages. That is transduction. And why I explained to you these life cycles, because these life cycles play an important role in taking host chromosomal DNA, bacterial chromosomal DNA, from one cell to another, from donor to recipient. Okay. So again, there are two types of transduction. One is generalized transduction, and another is specialized transduction. What is generalized transduction? Generalized transduction, as the name indicates, here the uh, the the donor DNA or the DNA from donor cell can be taken from anywhere, any part of the host chromosome, and transferred from donor to recipient. So it is generalized. I mean, what I'm trying to say, there is no specific regions of the host chromosome which goes from donor to recipient. The phage can take any part of the host chromosomal DNA and can transfer it to the recipient cell. So therefore, it is generalized transduction. Okay. In specialized transduction, what happens? The phage takes a very specific set of genes from host cell and then delivers it to the recipient cell. Okay, so that is specialized transduction. Okay, so the difference lies in the piece of DNA from the host chromosome or the donor cell DNA taken. In one case, it is taken from any part of the chromosome, and in another case, very specific part of the donor DNA is taken and transferred to recipient cell. Okay, so there are two types of transduction processes, generalized and specialized. We will learn what these, I mean, how these generalized and specialized transduction takes place. Okay, now if you see in this example what's going on, if suppose uh, this is the donor cell where the phage DNA is entering. We are talking about generalized transduction first. So in generalized transduction, what happens? See, the phage DNA is injected here, and this is the host chromosomal DNA, And if this is, suppose, Lu plus, we are taking as an example a marker gene. Okay. Now, as I explained in the lytic cycle, what happens? This phage DNA, where after entering, starts multiplying, and then it packages itself, But while doing so, what happens? Uh, I will explain to you how it happens that uh, at a very, very low frequency, the phage packages a part of the host chromosomal DNA, you know, along with its own means it will package its own DNA in 99% cases or even more, but at a very low percentage, it also packages the host chromosomal DNA like this. So when these progeny phages are released, it will have majority of them would be the normal phages, which will infect another cell, and some would have, instead of phage DNA, it will have the host chromosomal DNA, you know, host chromosomal DNA. And if this happens to be from the region which is having a Lu plus gene, and when this infects another cell which is Lu minus, here it is the recipient, you know, and when this DNA is released into the recipient cell, what happens? It recombines here and makes this recipient cell uh, from Lu minus to Lu plus like this, and Lu minus is degraded, you know. So it is allele replacement takes place here, you know, and you get a Lu plus transductant, you know. It is the transduction happening. This phage is generating or transferring this piece of DNA from here to here, which recombines, and you get a transductant produced. Okay. Now the question is uh, uh, how this happens, how the phage, instead of packaging its own DNA, it takes the host chromosome, part of host chromosomal DNA, and as I mentioned, it is generalized transduction. It can take any part of this host chromosome and package it here. Now, this phage is able to carry out the infection and injecting the DNA into the host cell, nothing else. And because it is carrying the bacterial DNA, the cell will not have any of the viral genes into it, only the bacterial DNA is packaged. Okay, so no harm done to this because there is no viral genes involved here. And then the recombination takes place, and one can look for the transductants on selection. Fine. So how this takes place? This mechanism is known, and we understand what goes in here. So actually, what happens? There is a nuclease we call P22 nuclease, which is phage coded nuclease, which cuts on a very specific site. We call it as PAC site, you know, this phage DNA, it cuts into a very specific site we call PAC site. And what is seen that the host chromosomal DNA also has similar sites, which are recognized by this nuclease, you know. So therefore, host chromosomal DNA is also cleaved off at different points, you know. But this is this looks like the PAC site of phage DNA. So, and it is called a pseudo PAC site. You know, it resembles with that. Another point I should make a mention here that this nuclease is very, very specific, and it recognizes its own PAC site of its own DNA to size that DNA to be packaged in the phage particles. You know, but at a very, very low frequency, it does recognize and cuts the host chromosomal DNA. And it is this part which gets packaged at a very, very low frequency in the virus particles. And therefore, it is called, and it can be in from any region of the chromosomal DNA, not any specific, wherever this PAC site is available at multiple places of the host chromosome, it is cleaved off and packaged. But this happens at a very, very low percentage, and we call it as generalized transduction. So you understand what's happening. The thing which I explained to you here, that this how this rare phage is generated, but this is cut by this P22 nucleus. it is cutting here also instead of trimming its own DNA, and then this gets packaged and recombined, and one can look for the transductants, Now, but then this doesn't serve our purpose from molecular genetics point of view. So what has been done that this P22 nuclease, as I mentioned, it's very specific to phage only, but then a mutation, a variant of this was created, uh, which is highly transducing. This P22HT we call, it's a variant of the normal phage, where what is done, this nuclease is mutated. And what happens after mutation? It loses its specificity, so it becomes less specific and starts identifying the chromosomal pseudopeptides, and therefore increases the frequency of the process which I mentioned earlier. And then also the progeny phages which are generated are more of more containing the bacterial chromosomal DNA as compared to the phage DNA. So what happens in specialized transduction? Here, as I mentioned earlier, a specific portion of the donor DNA is taken by phage and transferred from donor to recipient cell. So how this happens? Okay, so I will explain to you in a couple of slides how this happens. Okay, so look at this cartoon here. That this is the lambda phage chromosome. When it enters into the donor cell, it gets integrated into the host cell by process of recombination. There is a homologous region on the phage DNA or phage chromosomal DNA and the host chromosomal DNA, and it is integrated into this, in between two markers, for example, GAL plus and Bio plus, because we know that both these two markers are sitting close to each other. So there is a possibility that it gets integrated in between the two marker genes. Okay, now when so this will sit in the host chromosome generation after generation, but then when it enters into the lytic cycle, then what happens? It comes out of the host chromosome, and this is one way that a normal outlooping takes place, again between the two homologous regions, and it is excised out as normal phage, a normal phage chromosome, and it is packaged and released as progeny viruses. But as a rare event, this takes place. What is this here? That a loop between a part of phage DNA and the host chromosomal DNA. So here it takes place, and what you get as a consequence, instead of normal phage generating, it generates a defective lambda gal because it lacks some of these genes of the phage. It is defective. It cannot carry out its life cycle if this whole thing is delivered to a recipient cell in the next generation, because it lacks some of these genes. But instead, it is carrying some of the host chromosomal part of DNA and the genes which are located on that. In this case, for example, this GAL plus has come along with other markers also. We are taking it as just an example. Okay, so this phage chromosomal DNA is defective, and we call it as D-GAL phage chromosome. It is defective GAL phage. GAL, we are identifying that it is carrying a GAL gene or marker gene on it. Okay, like that, the specialization comes, as I mentioned, specialized transduction. It takes a specific region of the host chromosomal DNA. So depending on where it is getting integrated, in between which markers it gets integrated, and it finds a homologous region. And while coming out, it will take the nearby regions of its integration. Okay, fine. If you have understood this, let us see what is the consequence of this. So, if the recipient cell receives both of them, this and this normal phage, then it will follow the lytic cycle. But if a recipient cell receives or gets infected by the defective gal phage chromosome, I mean, this phage is capable of infecting the recipient cell, and injecting this type of DNA into the recipient cell. So what happens in this, that there is recombination between the incoming part of the donor DNA and the recipient bacterial chromosome. Okay, so if this recombines here, then and if the recipient cell is gal minus, then what will happen? You can see this minus is replaced by plus. Okay, and you can select this cell which is gal plus. It will be stable transductant produced. Okay, so here the gal plus gene is coming along with phage. So it is the phage which is bringing a gal plus allele or gene from donor to the recipient, which is lacking this marker gene. Okay, and making it from minus to plus. Okay, so this is what happens in the specialized transduction, and like that. I mean, depending on where the phage initially gets integrated, it takes the part of the host chromosome DNA and delivers into the recipient cell. Now, this which is after getting the gal minus region, since this is defective, it doesn't replicate. It's not able to replicate, and it is degraded. And you get a stable cell generated, which is gal plus. Okay, so it is the recombination or movement of DNA from one cell to another by way of these phages. So we have seen two ways. One is generalized type of transduction, specialized transduction. Okay, in both ways, a DNA moves from donor to recipient. Okay. Now, when it comes to gene mapping, actually, so here we look for co-transduction and the linkage process, what we saw in case of transformation. Okay, there also we look for co-transformation of the two markers, and here also we see the co-transduction of two marker genes and determine whether they are linked or not. As we saw in case of transformation, the two markers closer they are, the higher is the frequency of co-transformation. Same principle lies here for co-transduction. Closer the gene, higher the frequency of co-transduction. Okay, so for example, if you see in this cartoon, so you have, for example, let us see this. If we have a donor DNA moving from donor, which is gal plus and the bio plus, and different kinds of D gal or D bio or D gal bio plus phage have been generated. Depending upon what they are carrying, they may carry just a gal plus and bio minus, or they can have gal minus, or gal gene or the bio gene, or both the genes. So a phage can carry this portion alone from the donor DNA, this portion alone from the donor DNA, or both, because they are closely sitting. I told you earlier also that in a normal cell, both these markers sit close to each other, but there is a possibility of phage acquiring only this portion, or this portion. So when these phages infect the recipient cells and you look for transductants, you may get a gal plus, because the recipient cell is gal minus and bio minus. So if a phage, this type of phage infects this cell, it is going to replace gal minus with gal plus. So the recipient cell will become gal plus and bio minus. And similarly here, if this portion gets exchanged or recombined, you get a gal minus and bio plus. Okay. But if this whole region is taken from the donor cell, both gal and bio plus part is taken from the donor cell, and it recombines and replaces both gal and bio, you get a transductant which is gal plus and bio plus. And this is what we call co-transduction, wherein a cell is transduced by two marker genes, and both of them are coming together. Okay, and this is what is seen in when we go for gene mapping using this particular process for like transduction for gene mapping. We look for co-transduction, and what we mean by co-transduction is this: when the two marker genes move together and generate a double transductant. Okay, fine. Now, how this is then exploited? First, the generalized transduction. Let us see how the generalized transduction has been used for gene mapping. Okay, and the basis, as I mentioned, that closer the genes to each other, greater is the possibility of co-transduction. Okay, that is the presumption. Okay, if the two genes are transducing at a high frequency, it means they are sitting close to each other, or you may say they are linked also. Okay, so if we take an example of three marker genes A, B, C, and if we have a data like this, wherein A and B co-transduced at a frequency of 90%, A and C co-transduce at a frequency of 33%, and B and C co-transduce at a frequency of 32%. Okay? Now, each of these values have a statistical error of plus minus 2. So if you have this kind of data in an experiment of co-transduction of these three markers, what actually it indicates? See, these two values are very close to each other. So this indicates that B and C are sitting, B is closer to A. If you see, B is closer to A than C, because A and B has the maximum co-transduction frequency, 90%. So it means A and B are sitting close to each other as compared to C. C is slightly far away from them. Okay, and if we have to give a gene order, then one order can be ruled out straight. That is, A, C, B can be excluded. Okay, because we know that A and B are closer than to C. A and B are closer to each other than to C. So, this order can be excluded, but then what can be the other two orders? Then the order can be A, B, C or B, A, C. These two orders are possible. So how to decide that which one is the order, whether it is ABC or BAC? With this data, both these orders are consistent. We cannot decide that which order is correct. So to decide which order is correct, we go for a three-factor cross, between the donor and the recipient cell. Okay, so follow this experiment very carefully. So as I mentioned, we are presuming, see, in these kind of experiments, we put forth an hypothesis and expectations, and then we perform an experiment and we look at the data that which hypothesis is confirmed by the experimental data. So here you see we have considered two orders. One order is A, B, C. Another order is BAC. And this is what we want to decide, that which one of the two is the actual order, whether it is ABC or BAC. ACB is ruled out based on the previous experiment, So now, see, this is cross one. Here, what is done, you take a donor DNA, this red one is the donor DNA, and this is A plus, B minus, C plus. Okay. And the recipient DNA is A minus, B plus, C minus. In this order, you see B minus, A plus, C plus. Okay. This is BAC order we are taking, and the recipient is B plus, A minus, C minus. Okay. So this is one cross which was made and considering order 1, order 2. In another cross was a reciprocal cross. Okay. Reciprocal means, see, if this is B minus here in the donor DNA, and in reciprocal cross of this order, B is minus in the recipient DNA. Donor DNA is plus, and this is A minus, C minus. Here A plus, C plus, and B minus. Here A minus, C minus, and B plus. This is reciprocal cross. So donor, in reciprocal cross is what happens, the donor becomes the recipient. Here recipient is B plus, here donor is B plus. Okay, fine. And in this order, again, it is B minus, the donor, it is B plus, and A minus, C minus, here it is donor is A plus, C plus. Okay. And recipient is B minus, A plus, C plus. So this is the kind of crosses were made, and what was looked for? They look for A plus, B plus, C plus transductants. A plus, B plus transductants. Okay. Now let us see how one would get A plus, B plus, C plus transductants in all the four possible, in these four crosses. See here, if you see, this is A plus, B minus, C plus. So you need 4 crossovers, 1 at this, 2, 3, 4, to get A plus, B plus here, and C plus. Okay, so you need 4 crossovers. Okay, and here if you see, you get, to get A plus, B plus, C plus, A plus, then C plus, B plus, and then C plus. It means you require here only 2 crossover, or it's a product of double crossover. Okay, to get A plus, B plus in this order. If the order is this A plus, A, B, C, then in this situation, this cross must undergo 4 crossovers to give you A plus, B plus, C plus. And in this case, just double crossover. And if you see, if the order we considered BAC, and you are looking for A plus, B plus, C plus, then you can see just double crossover. B plus, A plus, C plus. One here, one here. Similarly, B plus, B minus, B plus, A minus, C minus, and this is A plus, C plus. So this is double crossover. Just double crossover. These are, and to get A plus, B plus, C plus here, and even here, you just need 2 crossovers. But here in this case, you need 4 crossovers. Okay. Now we have to decide which is the order, this one or this one. Now, the logic here is that the possibility of a cell going 4 crossovers is less than going 2 crossovers. Okay, so if this is a scientific fact. Probabilities of the DNA undergoing 4 crossover is less than 2 double crossovers. So therefore, if this is the order, and if you get more frequency of A plus B plus C plus as compared to this cross, it means this is the order. Among the two cross 1 and cross 2, if you see more of A plus B plus C plus in this as compared to this, then this is the order. But since see in cross 1 and cross 2, in this order, both of them require double crossover. So it means in this cross and in this cross, the number of transductants will be same, because the A plus B plus C plus transductants are the product of double crossover here, double crossover here. So there will not, you won't find any significant difference in the number of transductants you get in this cross and in this cross if the order is BAC. So just by comparing the number of transductants one receives in these two kinds of crosses, one can decide that out of A, B, C, these three markers, what is the order, whether it is ABC or BAC, So, and this is one of the objective for gene mapping, actually. You also look for is not only linkage among the marker genes, but also the order you can decide. Okay, so I think I'll stop here. And what I have tried to make you understand about transduction, that how this process, one of the parasexual behavior of prokaryotes, transfers DNA from donor to the recipient cell while completing its own life cycle. It is actually not doing for bacterial purposes, but this is the way bacteriophages complete their life cycle in the host cell. And while doing so, it so happens that it results into transfer of DNA from one cell to another, and a lot of information has been generated in molecular genetics by doing such studies, So with this, I conclude my sessions with you on parasexual behavior and genetic variability in prokaryotes, how the genetic recombinations generate the genetic variability, and of course, along with mutations, which are the key everywhere, whether in eukaryote or prokaryotes, to give you the variations which we see. So thank you very much for patient hearing. And if you have not subscribed, please do so quickly. Thank you very much.